Good evening, everyone. Welcome to the inaugural Corneal Stem Cell Club webinar. My name is Himal Kandal. I am a research fellow at the Corneal Research Group at the University of Sydney. I will be your moderator today. Before the event starts, we recognize and pay respect to the elders and communities, past, present, and emerging, of the lands that the University of Sydney's campuses stand on. For thousands of years, they have shared and exchanged knowledges across innumerable generations for the benefit of all. This is the agenda for this webinar, and I would like to point out some of the housekeeping rules. Kindly keep your audio off and video muted at all times. If you have any question, please post it on the Q&A box. We'll have a Q&A session after the presentations towards the end. During this webinar, we will launch two polls. The first will be launched before the first speaker and the second before the third speaker. At the end of the webinar, we will ask you to kindly complete a short feedback survey for this webinar. And within this week, in about a week, we will send a link of this webinar recording to all registrants. So our first speaker is Professor Nick D. Girolamo. He is a professor in the Faculty of Medicine and Health at the University of New South Wales, Sydney. He is a cell biologist with over 35 years of expertise and specialization in the corneal stem cells, who developed the first contact lens-based technique to deliver these cells to patients with severe ocular surface disease to improve their vision. Welcome, Nick.
Thank you. Okay, thank you, Himal, for the kind introduction. So I guess my role is to explain to you what corneal stem cells are, but I guess to begin the talk, I wanted to outline a few things, including what stem cells are, the types of stem cells and their use in treatment, stem cells for treating human corneal disease, and the importance of defining the corneal stem cell residence. So what are stem cells and what are their basic characteristics? Well, they possess lifelong self-renewal activity, which means they replenish the tissues and organs, our tissues and organs throughout life. They can change or differentiate into a diverse range of cells, and that speaks to their potency. They're protected by their residence, and in scientific terminology, that's called the niche. They're difficult to distinguish from neighboring or progeny or actual daughter cells, which makes them difficult to isolate. And the other, I guess, limitation is that there's no unique marker for these cells. What we do is we use a genetic signature to try and identify them. So the definition of a stem cell is the ability to undergo numerous cycles of cell division while remaining undifferentiated, but having the capacity to differentiate into specialized cells. So some facts and figures about stem cells. There are about 200 different types of specialized cells in the human body. And all cells in the body are derived from unspecialized stem cells. For example, the embryonic stem cell. The process of specialization is known as differentiation. And once a cell has undergone this process of specialization, it's difficult for it to revert or become another cell type. But when it does, and it can, so there are examples. For example, somatic or adult stem cells can do this, and pluripotent stem cells can do this. So when they actually revert, the process is called dedifferentiation or reprogramming. So I just wanted to highlight 4 types of stem cells, and there are probably a lot more in the body, and a lot more that we can derive. So the first type is the embryonic stem cell, actually derived from a fertilized egg, and the cell mass that is made from that fertilized egg from the blastocyst stage. So cells can be isolated from this blastocyst stage, and then they can be coached into pretty much every cell type in the body. The other type I wanted to mention is the mesenchymal stem cells. These cells are derived from cord blood, bone marrow, and fat tissue. They can be isolated and they can be directed to different cell types of the body, although slightly less so than the embryonic stem cell. The third type are the induced pluripotent stem cells. They can be derived from just about any cell of the body. So you can isolate any cell from skin, for example, or from the eye, and you can induce pluripotency by introducing some genes into these cells to make these induced pluripotent stem cells, which then can be differentiated into different types of cells. And the fourth type is the tissue-specific stem cell. These are the cells, or the adult cells, or the somatic cells that populate pretty much all the organs and tissues of our body. They can be isolated, although it's quite difficult because there are a few markers available, and they can be coached into the cell type from the tissue that they were derived from. So some applications of tissue-specific stem cells. Like I said, they're found pretty much everywhere throughout the human body. There are inner ear stem cells, and they're responsible for restoring acoustic function. There are pancreatic progenitor cells, which function as insulin-producing cells. There are intestinal progenitor cells, and we can infuse them into damaged intestines to repair them. There are myocardial stem cells to repair heart. There are all sorts of stem cells, including the ones that we find in the eye, and in particular in the cornea, which we call limbal or corneal limbal stem cells. And they've been transplanted in mice, in animals, and in humans to restore eye health and eye function. So just to get into these corneal stem cells and where they are and what they do. So the cornea is maintained in a healthy, transparent state because of the stem cells that reside in the limbus, which is this narrow annular region that separates the conjunctiva from the cornea. And it's populated by these stem cells, which their progeny or their daughter cells migrate from outer to inner in a centripetal manner to replace dead or dying cells throughout life. When these cells are depleted, or indeed the limbus is damaged, it's quite a catastrophic disease can develop called limbal stem cell deficiency. And it's characterized by this pannus of fibrovascular tissue that encroaches over the cornea and blocks vision. So I guess the gold standard treatment for total limbal stem cell deficiency, which is what this webinar is all about today, is a stem cell transplantation of sorts. So the importance of mapping the corneal stem cell residence, that being the limbus. So why is it important? And it's this annular region that is at the periphery of the cornea. So it's purported to be the regenerative zone of the cornea. It's the location of corneal stem cells. And I guess mapping its boundaries will make it easier to find and extract these cells. And that way we'd be able to create better quality, longer lasting stem cell grafts for patients in need. We've begun to map the landscape and I guess the confines of the limbus using two markers. Keratin 8, which stains conjunctival cells in green, and keratin 12, which stains corneal cells in red. So this is a representative image of a cornea which has been flattened, and it has the conjunctiva attached to the cornea per se, and it's been scored at four edges to facilitate its flattening. What you'll notice quite nicely is that all the conjunctival cells around the cornea stain green and all the corneal cells stain red. But there's this area in between the two different tissues that doesn't stain, and that's blown up in the image on the right. And you can see that the limbus is not staining for either of these two markers. So that's great for us because now we've been able to detect the genetic signature of all these green cells, the conjunctival cells, all the red cells, the corneal cells, and all the limbal cells, with the hope that we may be able to find better markers for these cells. So just to conclude this mini talk, I guess, we've mapped the corneal stem cell repository with decent markers to mark out the boundary of the limbus with keratin 8 and keratin 12. And we've obtained a genetic signature across the ocular surface to identify at least unique markers of stem cells. The goal now is to isolate corneal stem cells with these newly identified markers, determine their form and function in a dish, and hopefully conduct transplantation experiments in preclinical models and in patients with limbal stem cell deficiency. So just to conclude, I'd like to acknowledge the workers from my unit and my university and the funding agencies without which this work could not be conducted. Thanks for your attention, and I'll stop sharing.
Thank you, Nick, for the insightful talk. Our next speaker is Professor Stephanie Watson. Professor Watson is the head of ophthalmology specialty and head of corneal research group at Sydney Medical School, the University of Sydney. And she's also the head of corneal unit at Sydney Eye Hospital. And she leads the Safe Side Keratoconus Registry and Safe Side Dry Eye Registry as the chief investigator. Professor Watson is also the chair of Australian Vision Research, Vice Chair of New South Wales RANSCO, and Secretary of the Asia Pacific Ophthalmic Trauma Society. Over to you, Stephanie.
And so Professor Di Girolamo has given a very good background into corneal stem cells, and I'm now going to talk about some challenges in corneal stem cell therapy. And the reason why we need stem cell therapies because across the planet, there's about 2.2 billion people that are visually impaired. And this comes at an incredible cost, obviously, to the individuals and also to society, with over $600 billion burden for the Australian dollars alone. So it's these stem cells that hold promise. And that's because they're the building blocks of the body. They're a bit like Lego. You can make different tissues out of them. Now, we were fortunate to develop a technique where we were able to restore sight to patients with stem cell disease of their corneas using a contact lens, and this had medium to long-term success. But there remains many challenges in bringing treatments like this to the clinic so patients can benefit. So what are the challenges? Well, there's challenges for the eye and there's challenges for the patient. And so for the eye, the challenges include that we need to repair more than just one type of tissue in the eye, but also more than one type of tissue in the cornea. We need to restore what's known as the niche. This is the home of the stem cells. And so when we transplant a stem cell, we need to put it somewhere. And it's best if it goes into an environment that it will support its health. A lot of our current techniques, and when we developed our technique, relied on taking some tissue from the other good eye to grow it to then transplant back to a bad eye. But we need cell sources that don't rely on using healthy tissue from the patient's other eye, and also better ways to deliver stem cells to the eye. So in terms of repairing different tissues, the cornea is a bit like a sandwich. It's got a number of layers. The top layer is this layer of epithelial cells, and that's what is replenished by these limbal stem cells and replenished in our technique with the contact lens. But the meat of the cornea, the sandwich, most of it is layers of collagen, one on top of the other. And then on the very inner surface of the cornea, you've got these red cells, which are known as endothelial cells, and they work by pumping fluid out of the cornea because the eye's fluid that's inside is constantly trying to enter the cornea. Now, there's a condition called keratoconus, and it's a bit like when the sandwich layers slip on each other because you don't have enough toothpicks in, and the cornea can become stretched out. And as well as developing techniques to repair the limbal stem cells in the surface of the cornea, there's now an interest in developing stem cell techniques for conditions such as keratoconus. And in one study, stem cells from fat tissue were actually taken and transformed and then implanted into the cornea of patients with keratoconus to thicken them up, and there was some improvement in vision. Now, what about the challenges in restoring the niche? Well, the niche, or the home for the stem cells, is very important because it contains the signals that keep the stem cells behaving as stem cells. And in many conditions, the niche can be damaged. which means that if you give back stem cells, they may not necessarily behave in the right way for the long term. We discovered a factor, vitronectin, that was able to support in culture limbal stem cell function. And so treatments need to be developed, and we need to find out more about this niche in order to provide the stem cells with an appropriate home. Now, cell sources, we need new cell sources. And one of the cell sources that's become very popular for stem cell therapies is mesenchymal stem cells. And these are like, rather than epithelial stem cells, these are like more like the fibrous tissue type of stem cells. And they've been found to be able to repair and renew connective tissues. They're also anti-inflammatory, anti-fibrotic, and they do have some immunosuppressive factors as well. There's a new type of stem cell as well called an induced pluripotent stem cell, which sounds very fancy, but we call them iPS cells. And these cells are reprogrammed cells. So you can take a cell from skin and then you can make it into a corneal cell. As well as cells, there's now work on creating little mini organs and just using even components of the cells. But there's still much more work to be done. So the future directions in terms of stem cell sources is that we need to be providing high quality stem cell sources for use in procedures such as transplantation that can be used for many people, that don't just rely on a biopsy from the patient's good eye, and that these cells need to retain their stemness. And one way of doing this might be to create stem cell banks, where we have banks of tissue available that can then be provided to patients in need. But for all of these cell sources, we need to focus on as well avoiding rejection. If we transplant pieces of stem cell, not just the cells, to a patient's eye, there are risks that that tissue can be rejected. And if that happens, then the stem cells won't work to repair the eye. And people are working on different ways to try to avoid rejection. But again, there's more research needed to solve these problems. For cell delivery, we delivered the cells with a contact lens, which was very practical. Other groups are looking at a range of different tissues for delivery. One of the ones that has been used, and it's shown in this picture, is an amniotic membrane, and that's taken from placenta. And it's a bit like a biological band-aid. It contains lots of growth factors, but it does need to be sutured on, and it does dissolve over time. We are working in our group on some new solutions for cell delivery. What about the patient? What are the challenges for the patients with stem cell therapies? Well, the first one is really treatments that are able to restore sight. But it's no good having a treatment that works, but that patients can't access. So another challenge is accessing the therapies. And then as we go on as new treatments reach the clinic, we need to be able to evaluate them to know that they work and also avoid complications. So in limbal stem cell deficiency, which you've heard about, it causes scarring, loss of vision, and it typically affects a working age population with a leading cause of chemical injury. And we can't treat it with standard grafting. But there's many, many more conditions, corneal conditions, and also conditions elsewhere in the eye that require restored vision from stem cell therapies. And this causes issues as well with access to treatments. When we performed this study in 2013, we found that one of the limitations for access to treatments that might potentially restore sight is actually a lack of funding, and that's both for bringing the treatments to the clinic and for the research to support them. Now, once a therapy is in the clinic, we need to evaluate that therapy to know that it works. And we do clinical trials to make sure a therapy is safe and works, and to bring it to the clinic. But once a treatment is in the clinic, we then need a way of monitoring it long term, and we can do this with registries. And this provides us with real world data and real world patients, because in clinical trials, the included patients have to meet very strict criteria. And the trials are often short, whereas with registries, we can get long-term follow-up. This is the Save Sight keratoconus registry. We have one for dry eye as well, and it would be ideal to create one for corneal stem cell therapies as they become available in the clinic, as it will enable us to track the long-term outcomes and also what we know as patient-reported outcomes, which is the outcomes that matter most to patients. And they've been increasingly recognised, these patient-reported outcomes, as important for regulatory approval for treatments and also for their follow-up to make sure that we are delivering treatments of benefit to patients. Now, we want to make sure that with our treatments that we avoid complications. And it has been written that some stem cell medicine is a bit like the Wild West. And some of the concepts that are being employed in treatments that are available aren't necessarily scientific. In this publication from the United States, it highlights an unfortunate stem cell therapy where fat-type cells were injected into the eye and led to blindness. So we created these resources for patients and the profession seeking to know more about stem cell therapies and whether they're safe. And this flyer, which highlights 5 questions that a patient can ask their practitioner if they're thinking about a stem cell therapy. It's important to know whether the therapy is regulated, what evidence is for its success, and what will happen in the long term. Just lastly, there was the Lancet Commission on Stem Cells, and it came to some conclusions which I think still hold, in that stem cells hold promise for therapy, particularly corneal stem cells, as the cornea is the most accessible place in the eye and a good place to test and develop stem cell therapies. But importantly, the reality of what's developed must match the expectations. We are still awaiting a range of stem cell therapies to reach the clinic. We have lots of research to support the directions we should head in. And we must always keep developing therapies that will be cost effective so patients can benefit from them. We need to make sure that any stem cell treatments occur in regulated trials and there's registries to track their progress in the long term. And while we do all this, we need to engage patients and society to make sure that stem cell therapy delivers on its promise. Thank you very much, and thank you again.
Moving on to next presentation. Our next speaker is Dr. Maria Cabrera August. She is a research associate at the Corneal Research Group at the University of Sydney. Her research focuses on educating the community on prevention and treatment of corneal conditions. Over to you, Maria.
Thank you, Himal. Gonna share my screen. Well, hi everyone, good evening. Thank you, Himal, for the introduction and the opportunity to present today on patient awareness on ocular stem cell therapies. Corneal conditions can benefit from stem cell therapies, for example, for limbal stem cell deficiency, which burdens patients of working age with devastating effects on vision, with ectodermal dysplasia as a rare cause. And also for keratoconus, which typically onsets in the youth and progressively reduces vision. And the aim of this presentation is to show you the results of a survey that we conducted last year to know more about the awareness among patients with keratoconus, limbal stem cell deficiency, and ectodermal dysplasia of stem cell therapies as a potential treatment for their conditions. We developed an online survey, and we circulated this survey among members of the Keratoconus Australia and Australian Ectodermal Dysplasia Support Group. The respondents were over 18 years of age. We did this from July to September last year, and we collected the patient details, patient medical history, awareness of an experience of use of stem cell therapies, and concerns of use of these therapies. We obtained 404 responses, mostly were female respondents between 46 to 60 years of age, who lived in Victoria, and they mainly came from the Keratoconus Australia support group, Hence, well, the main diagnosis was keratoconus, and the time since diagnosis was between 21 to 30 years. The main concern that they expressed was about safety of these therapies, and around 85% were not aware of any stem cell therapies in the standard practice, and around the same were not aware of clinical trials investigating stem cell therapies. When analyzing this survey, we identified 4 main themes. One of those were the safety and cost concerns. So I brought some quotes from the respondents. For example, one male respondent said, oh, I open quote, as with all therapies, time to prove safety and efficacy. And a female respondent said, it feels like stem cell treatments must be miles away from either being invested in or affordable for keratoconus patients. Another theme was the age-related concerns. One female patient said, I open quote, I'm almost 70 years old and have good vision in one eye, and adequate vision in one, the other eye with a scleral lens. However, I do worry about my future as I get older and have to manage these lenses. And I don't know if stem cell therapy is even an option for older people. Another female patient said, stem cell therapy sounds amazing for the next generation with corneal disease, but at my age of 53, with advanced keratoconus, is most likely too late to be of benefit to me. Another theme was the lack of knowledge of these therapies. One male patient said, I've had 5 transplants and no doctor have ever mentioned this to me, so maybe it doesn't apply, but I would like to know more if it could help me. And the last theme was the interest in learning more about the corneal stem cell therapies. One female respondent said, I have never heard of this type of therapy in relation to keratoconus, and I'm going to start researching now and asking my specialist about it. And another male respondent said, I would be highly interested in knowing more about stem cell research and treatments in this area. I have basic understanding of stem cell research and applications, and I would be interested in being included in any trials of such treatment. Finally, I would like to give you 3 take-home messages. First, that most of the respondents have had the corneal condition between 10 to 30 years. They were not aware of stem cell therapies for the treatment of keratoconus or limbal stem cell deficiency. And the main themes from this survey were the safety, cost, age-related lack of knowledge, and interest in learning more about these therapies. And these findings will inform educational resources for patients awaiting these therapies. And finally, I would like to acknowledge Keratoconus Australia and the Australian Ectodermal Dysplasia Support Group to help us circulating these surveys among their members. Thank you.
Thank you, Maria, for presenting the interesting data, highlighting the need for more patient-centered research in this area. Our next speaker is Professor Neil Ligali. He is a professor of experimental ophthalmology at Linköping University in Sweden, where he leads a research group with a focus on understanding corneal diseases and developing new regenerative therapies. Welcome, Neil.
Thank you so much, Himal, and I'd like to thank Professor Stephanie Watson and Mojtay Abbasi for the kind invitation to speak to you today. I'm going to share my screen. So my name is Neil Ligali. I'm at Linköping University in Sweden, and good morning to everyone. Today, I'll be talking a little bit about how we use novel models of LSCD for therapy development. So limbal stem cell deficiency, as you've heard in the previous excellent talks, can be acquired or traumatic, but it can also be congenital or as a result of autoimmune diseases. And it can be a partial or a total stem cell deficiency. And as previously explained, the limbus is a region just between the clear cornea and the conjunctiva, covering the white part of the eye, where there are specialized cells, including stem cells, that maintain the corneal epithelium for its transparency. However, in limbal stem cell deficiency, there's a disruption of these limbal stem cells, and they can no longer produce the transparent corneal epithelial cells. And this is often accompanied by inflammation, a loss of nerves, and vessels that invade the cornea. You can see this here in the case of total limbal stem cell deficiency. So current treatments are surgical, and they have led to variable levels of successes, as we can also see in the polls that were done. We are interested in models of limbal stem cell deficiency. And why do we need these experimental models? Well, first of all, we'd like to understand better the mechanisms behind LSCD and what leads to it really at a genetic and molecular level. And the models are a way we can actually examine the tissue in a way that we can't in patients. And secondly, the models allow us to evaluate new potential therapies for limbal stem cell deficiency. So what are the characteristics of good models? First of all, they should mimic the human disease. They should give us the same clinical picture that we see in patients. Also, the development of the disease should have approximately the same timeline as in the clinical case, as well as we should see the same levels of severity in our model as patients experience. Also, the model should be reproducible. So each time it's used, it should give us the same kind of result. It should enable us to apply therapy, so either be it a transplantation or a cell therapy or a drug, that we can apply it in the same way in the model as in patients. And finally, the model should be easy to use in the laboratory and easy to understand and assess to see if our potential therapies are effective or not. I'm going to talk about congenital limbal stem cell deficiency. So this is LSCD that's caused by genetic mutations in utero that after birth lead to the development of LSCD over time. There are two genes that we're principally interested in. The first is PAX6, which is a major gene that controls the development of the eye in utero. And another gene called TP63, and this is responsible for development of ectodermal tissues. So our skin, for example, the hair, teeth, and also the cornea. And importantly, both of these genes are what is known as transcription factors. So these genes produce proteins that then bind to DNA and modify the function of DNA and the expression of a host of other genes. And these congenital cases of LSCD can either be sporadic, so they can arise newly for the first time in a family, or they can be due to hereditary mutation in these genes, so that the child can inherit this from one parent, and it's often one mutated copy of the gene that's inherited. And importantly, these congenital cases lead to stem cell deficiency in both eyes, and often it's just as severe in both eyes. So in these cases, the condition can very quickly lead to blindness. In terms of the first gene that I mentioned, PAX6, mutation in that gene leads to a disease called aniridia, which is characterized by underdevelopment of the eye structures. And also it leads to a progressive limbal stem cell deficiency that usually starts in the 20s and then progresses from there. Again, it affects both eyes, and there's generally a very poor prognosis because the eye is very inflamed, and there are no really good treatments for this today. So we're using a model of PAX6 mutation in mice that mimics this human disease timeline for aniridia. And this was first developed by Elizabeth Simpson in Vancouver, and it's then worked on by our PhD student, Dina Javitjan in Sweden. And this is a picture of the model, the eye over time. So you can see over several months that the eye becomes more and more opaque and the blood vessels start to grow further and further into the eye. So this is progression of the disease. And we can compare this to the clinical picture that you can see in a patient, and it looks very similar to the mouse model. So we use this model for testing new therapies, potential new drugs or cell therapies, and with the aim to either slow or stop the progression of the disease. And the other gene I mentioned was the TP63 mutation, and this results in a disease called EEC syndrome, which is an ectodermal dysplasia, which affects the skin, but also can affect the eye. And when it affects the eye, there are problems with production of the tear fluid, as well as inflammation in the eye, scars, and limbal stem cell deficiency. So we also are using a mouse model of the TP63 mutation that mimics, again, the human disease. This was developed by Katerina Misero and then modified by Ruby Shalom-Feuerstein. It's genetically engineered so that the mutation does not occur in the skin, but just in the eye. And here's a picture of patients with this mutation. And you can see here by the arrows, you can see the limbal stem cell deficiency starting in this patient. And if we compare this to the mouse model, we can see that over time, this is showing from 2 to 18 months, that the condition of the eye becomes progressively worse. And when we look at the cornea, we can see that there's more and more blood vessels and it gradually loses its transparency. So these models produce the what we see in the clinic in limbal stem cell deficiency, but on the time scale of several months to up to a year. The problem then becomes that it takes several months to a year for one experiment, and often we have to repeat experiments because we want to try different dose or concentration of drugs and optimize therapy. So this could take several years. So we need a way to accelerate the limbal stem cell deficiency. So what we do is we make a small central wound, just a superficial wound in the epithelium, that in the normal cornea would heal very quickly and restore transparency. But in LSCD, it advances the disease in a very short time. So this was done by 2 master students in my lab, where we used this device to create a very precise wound in the central cornea of the mouse, 1 millimeter, where we just remove the epithelium. And in the normal case, after a week, this heals very nicely. But in the lower pictures, you can see in the case of LCD, after 4 weeks, it advances the disease to an advanced stage with vessels covering the entire eye. So this is a way to speed up the LSD. So in conclusion, we use experimental models to help us better understand LSCD, to find out what's going on at the molecular level, and to uncover new potential therapeutic targets. And then we use the models to test candidate therapies so that we only proceed with those that are showing positive effect in our models, and this saves us time and resources. So with positive data, we can then apply for permission for clinical studies and ensure that only the best therapy candidates eventually get used for patients. So with that, I'd like to thank you very much for your attention.
Thank you, Neil, for the insightful talk. Now I would like to move on to the next presentation. Next speaker is Dr. Mozdeh Abbasi. She is a postdoctoral research fellow at the University of Sydney, specializing in corneal biology with a focus on developing innovative therapies to prevent vision loss. Over to you, Mozdeh.
So I would like to talk about corneal stem cell therapy for limbal stem cell deficiency, research, communication, and patient impact. So due to time shortage, and as we heard about the disease, the causes behind the disease, and what happens to the eye after limbal stem cell deficiency condition, I will skip my slide 1 and also my slide 2, which we know that the current treatment for limbal stem cell deficiency, also stem cell transplantation. And the new approaches like using new sources for stem cells, for example, oral mucosal epithelial cell. But as we heard earlier, there are challenges, like, for example, patients need long-term immunosuppression, and long-term results are not clear. We might have a lack of donors, and there is a risk of infection and damage to the healthy eye, unfortunately. And we don't have any good solution for the genetic issues. That's why our group in University of Sydney and the Centenary Institute are working on a new method for 2 areas. For example, that first, we want to improve our only one eye, and we are also developing new approaches for patients who have bilateral condition, which means damage of 2 eyes. We are using a new method that is actually growing the cells on contact lenses. And why contact lenses? Because they are affordable, widely available, they are transparent. So following up the patients to see how the healing is going on is quite easy. And more importantly, we have shown that the cells, when they are growing on the contact lenses, can maintain their stemness, their properties. Here you can appreciate that the proteins that are exist in stem cells under normal condition, they have similar patterns when they are also growing on contact lenses. And on bilateral condition, which both eyes are damaged, we are developing a new method by taking blood samples from the patients. And we are actually reprogramming the blood cells into induced pluripotent stem cells. And then we differentiate them into, because these cells can be leaded to any type of cells in body. And we are differentiating them to limbal epithelial stem cells, growing them on contact lenses, and transplant it back on the patient's eye to restore sight. This is what we are working on right now. After we are developing our stem cells on the contact lenses, we would like to test them by using preclinical model. Here you can appreciate a preclinical model that we are currently using. And this colorful spot shows the stem cells that when we grow the stem cells and we want on the contact lenses and we want to check them on this preclinical model, we can track these colorful, which are showing the stem cells, to see how the regeneration is going on. And this image was actually go beyond science, and it won the prize at Art Meets Science competition. And finally, I would like to talk about the importance of public communication. So between, of course, the researchers and the public community. We all know that science doesn't only happen in the lab. And for the research to be truly become treatment, we must share it, listen to patients' experience, and we would also like to show and explain our research to them. So this research communication is quite important between the researchers, the patients, the public community, and even policymakers. For example, a few months ago, I had the chance of attending an event that is called Science Meets Parliament. This is an event that actually happens once a year, and the researchers and the scientists have this chance to meet in person the policymakers, and they can talk about their researchers and the challenges they meet while they are doing their works. And I found how important it is, because it really can influence the policymakers' decision, for example, on healthcare priorities and even on research funding. And it's important, so this type of communication, because it builds trust between us and the patients, and it actually bridges the gap that normally exists between the research in the lab and the general community. And finally, I would like to have 3 take-home messages. So our goal is restoring vision and improving the quality of life of patients with limbal stem cell deficiency by developing our new method on contact lenses. And fortunately, corneal stem cell therapy holds promising future. And finally, communication between researchers and general community plays an important role in translating research into real-world impact. And thank you, everyone.
Our next speaker is Amanda Craze. Amanda is the Associate Director of Development for the Faculty of Medicine and Health at the University of Sydney. Welcome, Amanda.
Thank you, Himal. Thank you, everyone. Pleasure to be here today. I'm just going to share my slides. So yes, really enjoy speaking to people about philanthropy, and I'd like to share some insights into the role of philanthropy supporting corneal disease research today. So in Australia, eye research receives funding through a combination of public and private sources, such as individual donations, family foundations, and community grants. Public funding comes from government agencies such as the National Health and Medical Research Council and the Medical Research Future Fund. These are government schemes providing grants and fellowships to support researchers, and the NHMRC and MRFF only funds approximately 6 to 10% of all grant applications received each year. So you can see from these averages that the success rate for Australian researchers nationally is an extremely competitive process with a low success rate, even if their research is really, really important. This can lead to critical research not receiving funding and further reducing the opportunity for treatment or a cure. So the challenges that researchers face is that it continues to be difficult to secure dedicated, prioritised funding for eye research, yet conditions such as corneal diseases continue to impact an increasing number of individuals globally with limited therapeutic options or cures. There are limited funding sources available for research, and funding organisations must allocate these resources across many medical conditions and scientific disciplines, meaning corneal disease research is competing with other important areas of medical research for funding. The cost of research can be a significant challenge too. Cutting-edge research, especially clinical trials or genetic studies, and developing therapies can be expensive, and a lack of awareness can translate into less public support, which can actually mean less funding. So how can philanthropy help? Public advocacy and education are crucial for supporting research funding. Donations and support from our donors play a hugely important role, and it's vital to bridge this current funding gap. We have to secure long-term funding sources, so this helps to ensure that we attract and retain top research talent. Researchers prioritise projects based on the greatest need and impact, like you've seen in this presentation today. Long-term research is also supported and then enabling deeper and more meaningful data collection, and secure funding accelerates progress, patient access to therapies, and enhances the global patient impact. So no matter how large or small your donation, your support makes a difference now and into the future. There are various ways to support the Corneal Research Group at the SafeSight Institute, by either a single or regular monthly donation, donating through your workplace giving program, considering a gift in your will to the Corneal Disease Group at the SafeSight Institute is also a meaningful way to contribute to future eye disease research. And then if you have capacity, a larger philanthropic gift can help secure sustainable funding to tackle some of our biggest eye disease challenges. Every dollar donated to the SafeSight Institute supports advances in diagnosis, treatments, and brings us closer to a cure, and also benefits our global community through this incredible research. So thank you for your time. If anyone would like to learn more about how you can support the research showcase today by our researchers, please get in touch via my details on this slide. And you can also scan this QR code if you would like to make a donation today. So thank you.
Thank you, Amanda, for sharing the important information on how to support our research. We are coming towards the end of today's presentations, and on the slide you can see we are present on different social media. So to keep updated with the research that we are conducting, please follow us on these social media. Now we'll have a brief Q&A session where we have run slightly over the time, but we'll have a brief Q&A session. And I would like to ask all the presenters to unmute and turn the videos on. I would like to start the question with Nick. First question: how many corneal stem cells do we have And do they change in number as we age? Thanks, Nick.
Okay, thank you. Thanks for the question. Well, I guess no one really knows for certain how many we actually have or how many we're born with, and whether that number remains with us for the rest of our life. The issue is, as I mentioned in my talk, that there's no unifying marker or super specific biomarker for these cells. But I mean, if I had to put a figure on there would be in the vicinity of potentially hundreds to thousands, but not tens of thousands to millions. And going from what we've done in the past and what others have published, I think it's possible that they preferentially populate different sectors of the limbus. As to why, I don't think anyone really knows, but maybe different sites house different stem cells for different functions. And to answer your question about how many do we actually need for a successful transplant, again, no one really knows. It's a really interesting question that's been bugging me for ages. I think it hasn't been appropriately explored. There is one or two publications, if I recall correctly, from an Italian group that have suggested that a graft that contains at least 10% of donor cells that are stem cells, those grafts do better than when the stem cell population is less than 10%. I hope
Thanks,
I've answered that question.
Thank you, Nick. That's great. And we have some questions in the Q&A box as well. There's a question here. Probably Neil can start answering this. Can we make an in vitro model for mimicking LSCD?
Is that question for me?
For Neil.
Oh, for Neil, yeah.
Yeah.
Sorry, I
Can
can't
we
see the questions here. What was the question?
can we make an in vitro model for mimicking
yes.
LSCD?
Okay. Yes, there are in vitro models for LSCD, and these involve limbal stem cell, often limbal stem cell lines. So they're immortalized cells, although primary limbal stem cells taken from, for example, cadaver eyes can be used, or from animal sources. So yes, these do exist and they are used in research.
Thank you, Neil. My next question is to Stephanie. Is there any approved corneal stem cell therapies for LSCD or keratoconus in Australia or elsewhere?
So one of the world's first therapies, stem cell therapy approved was actually in Europe for limbal stem cell failure. And that was approved in the European Union. And that involved the culture of stem cells to the eye to treat the deficiency. There's no approved therapies in Australia at present. There are some surgical procedures and some medical treatments depending on whether the stem cell is partial and whether it affects one or both eyes. So there are treatments that can help people with stem cell therapies, and it depends on the state of the condition. But the treatments for total stem cell deficiency in terms of stem cell cultures aren't yet approved here.
Thank you, Stephanie. My next question is to Mosde. What scientific developments do you expect to see in the future of corneal disease research and treatment?
Oh, well, actually, all type of the treatment might have their own side effects. But what we are doing in our research in our lab, we are trying to be strict about all the conditions, regulatory things. So in terms of using the cells for stem cell therapy, we are doing our best. However, there might be some, for example, inflammation or irritation, which I think they are just by normal eye drops, they can be removed easily.
I have one question to Maria now, the last question for today. What are the next steps with the survey results? What is the next step?
Okay, so in our project, so the first part was to do the survey, and then now we are conducting interviews to patients with limbal stem cell deficiency about their quality of life. So once we finish those interviews, with those results plus the survey results, we're gonna create some educational resources for patients. And then, for example, PDFs, we can send them via email, brochures that we can hand at the clinic. We are gonna do some podcast. We have created a podcast here at the group. It's called the Sydney Eye Podcast. So we're gonna create a new season for next year. So we're gonna do a podcast and potentially videos, animated videos, also to share the knowledge among the community.
I've just posted a link to the feedback survey, so that will help us to plan the future events. Thank you, everyone. Now, we are at the end of today's webinar, and I would like to thank our speakers for the amazing presentations today, and the audience for their questions, and for joining us today. Thanks for joining us, and have a great evening.
Thank you.
Thank you.